In order for us to understand how neurons work, we have to look at ion channels that are found in the plasma membrane of neurons. These ion channels are proteins that have openings in them, and when these ion channels are open, ions will move down their electrochemical gradient. When we talk about the electrochemical gradient, we have to consider two things that are going to determine which direction an ion is going to move. One force that will influence the direction of the flow of an ion from one side of the cell membrane to the other side of the cell membrane is the chemical gradient. The chemical gradient is a force that will tend to want to move the ion from a place of high concentration to a place of low concentration. An electrical gradient is a different force. Positively charged cations will want to move towards a negatively charged area, and negatively charged anions will want to move toward a positively charged area. So these two forces, the chemical gradient and the electrical gradient, influence the direction that an ion will move. As ions move through these ion channels that are found in the plasma membrane, they will create a flow of an electrical current, and that can change the membrane potential. In other words, that can change the amount of charge on one side of the membrane versus the other side of the cell membrane. Let's now take a look at ion channels. This is what an ion channel looks like. As you can see, it has a gate, and these gates will determine whether an ion will flow through it or not. Let's now take a look at specific types of ion channels. In this picture, we will first look at leakage, or otherwise known as non-gated channels. These type of channels randomly alternate between the open and closed position. Many textbooks will say that these channels are always open, and for all practical purposes we can kind of think of them that way. In other words, they are constantly allowing ions to flow through them, and the ions will flow following the electrochemical gradient. Nerve cells have more potassium leakage channels than sodium leakage channels. This will be significant when we talk about neurophysiology, so simply remember this important fact, that there are more potassium leakage channels than sodium leakage channels. And as a result of there being more potassium leakage channels, the membrane permeability to potassium is higher than it is to sodium. Again, remember this because this will be important later when we discuss neurophysiology. Another type of channel is called a ligand-gated channel. These kinds of channels will open and close in response to a stimulus. The stimulus will be something that's a chemical such as acetylcholine, as we can see here. In other words, when acetylcholine is released from a motor neuron in order to stimulate a muscle fiber, the acetylcholine attaches to receptors that are in the motor end plate of the muscle fiber. The receptors are in fact these kinds of channels. When the acetylcholine attaches to the receptor, or the ligand-gated channel, then ions will flow through. This results in neuron excitability, or in the case of muscles, muscle excitability. Mechanically gated ion channels respond to mechanical vibration or pressure. These would be the kinds of ion channels that would be part of sensory receptors that pick up things like touch in your skin. And lastly, a very important type of ion channel when discussing neuron physiology is called a voltage-gated channel. These kinds of channels will open and close in response to a direct change in the membrane potential. In other words, whenever there is a change in the amount of electrical charge on the two sides of a plasma membrane, this will affect the voltage-gated channel and cause it to either open or close.